All right. Hi, everybody. Oh, look at that response. I hope that you are all doing well. Remember that you have a VDJ problem set due on Monday next week. You know how to do most of it, but not all of it, but you should know how to do all of it by the end of today. On Friday, we're going to be talking about some details with regard to techniques that will then help you for the other techniques assignment. So just be aware of all of that stuff that is upcoming. I think happily, not totally happily, but no one came to my office hours today, which meant I got to spend the entire time troubleshooting tech. So I think I have everything worked out to do some snazzy stuff today. And I figured out why my mic wouldn't work on Monday. So I think I got it all figured out. So today we're going to continue talking about the process of generating antibody diversity. AKA VDJ recombination that we started on Monday. And so here, if you remember, is our overall process of VDJ recombination. We have our mini gene segments. In this case, we have a light chain because there's only V and J. If there was VD and J, we would have a heavy chain. And you can see that each individual B cell will choose a V region and a J region and paste them together and cut out the DNA in between to make this new coding sequence that codes for the receptor. You will see your textbook here calls it somatic recombination. Somatic just means a body cell, like not an egg or a sperm. So the idea is that, oh my gosh, you're getting DNA damage, DNA changes in an actual body cell. Shocker. And we started talking a little bit about the process of how we were going to have the DNA cut at those individual gene segments and then pasted back together. And I introduced you to the idea of RSSs, or recombination signal sequences, which are these unique sequences that are adjacent to our V's, D's, and J's that our enzymes are going to actually grab onto in order to find two segments that it's going to recombine together. So the basic idea is that the enzyme that we're going to see is going to, pick a V, pick a D, bind to them, and eventually recombine them. And that binding is going to happen at these recombination signal sequences. There are two kinds. They differ in the length of the spacer in between two conserved sequences. So one has a 23 base pair spacer, one has a 12 base pair spacer. You can see they're drawn here as either brown or yellow. And we talked about the 12-23 rule, that you can only join together two segments that have a 12 with a 23. You can't put together two 12s, you can't put together two 23s. And you can see our light chain. We've got one kind on V, one kind on J. Yes, it differs between the two light chains. No, I don't care. And in the heavy chain for B cells, you can see we've got the same one. In this case, it's 12 on D. We've got 23 on J. We've got 23 on V. And so we saw this, this process. One other thing I will just mention briefly, if this confuses you, then you can ignore it. But there are a couple places where it sometimes comes up in questions, or sometimes will come up in literature. Like, I'm not going to ask you questions about it, but sometimes students will ask me questions, is what I meant. You can see that this shows the two different RSSs that are sort of going to recombine with each other, pointing towards each other. The arrow actually could point the other way. Like, you could actually have a brown one over here. I'm never going to draw it for you, but that is actually possible. And technically, when that happens, the intervening DNA is not deleted, it's flipped over. So it's called inversional joining instead of deletional joining. There are a few places where people will use it in experiments where they'll try to flip over a piece of DNA. But I'm not going to sort of go into that detail and confuse us with that additional detail. But sometimes students will be like, you know, do they have to be like on these, in these intervening, in the middle parts? And no, you can actually have a RSS over here, but then you end up with a different way for VDJ to work out. And I'm not going to go into a ton of detail on that. Here you can see that, again, in a version from your textbook, instead of drawing the RSS as a triangle, they're actually showing you the conserved sequences, the heptamer H, nonamer N, and then the spacer. And you can see that those two are brought together. So this would be a really big triangle like that. And this would be a really big triangle like that. So those would be the triangles. The triangle includes the heptamer, spacer, and nonamer. And so you can see that all of this DNA in between those signals is going to get deleted. So in the end, if I draw it here as well, oh, this is going to be difficult drawing for my drawing levels. That's supposed to include the N, but I don't know how to draw a triangle that also includes the bends. So we'll just do it like that. So you can see that the RSS is included in the deleted DNA, and that we are putting together the gene segments without that RSS in between. And like I said, yes, the triangle should include the N, but my art skills are just not that good. And you can see all of that DNA that was in between got deleted. These are some of the proteins that I mentioned are involved in VDJ recombination. I'm specifically going to be calling out RAG1, RAG2, TDT, and Artemis. And most everything else involved here, I will just talk about as like the polymerase, the ligase, things like that. Many of them are also involved in things like DNA repair. So there's a bunch of overlap here with DNA repair processes. In fact, technically Artemis, it was discovered by the immunologist, as I will explain today, but technically it actually also functions in DNA repair, we now know. The only one of these that are special for lymphocytes are RAG1 and RAG2 and TDT. And when I say special for lymphocytes, I mean that they are the only ones that are expressed only in lymphocytes. So every cell of your body has the genes for RAG1, RAG2, and TDT, but every cell in your body doesn't transcribe those genes and translate the proteins. So the only cells that actually have these proteins, as we're going to see over and over today, this is also a hint for your homework. There's one question people sometimes answer very philosophically, and I do not mean it philosophically. I mean thinking about enzymes and what enzymes are present in terms of being transcribed and translated. Only lymphocytes, only the cells that do VDJ recombination transcribe and translate RAG1, RAG2, and TDT. That's all. I could also say only lymphocytes express those. So those are like, those top two are really unique lymphocyte proteins. Everything else on here is actually in every cell type taking part in DNA repair. So we're going to go through this VDJ recombination process here. So what you can see is that the first thing that has to happen is we have to have some protein that's going to recognize and bind the RSS. So the first thing we have to do is recognize the RSS and bind to it. And that if I'm being super technical, is done by a protein called RAG1. There are actually two proteins, RAG1 and RAG2, that are different enzymes that do a lot of work together. They usually kind of spend a lot of time together as part of a complex. That's why they're shown here as a big blue blob together. Technically, they are two enzymes encoded by two genes. And the paper that we're going to be talking about on Friday next week is about the discovery of RAG2. Though it does mention some stuff about how RAG1 had previously been discovered. So RAG1 actually is the recognizer of the pair. And so you can see RAG1 has picked this particular V. I don't know what number it is because they don't tell us. And has picked this particular J. So this must be a light chain. They don't tell us what number it is either. And RAG1 has actually bound to the RSSs. And you can see RAG1 actually kind of bends the DNA around to get those RSSs next to each other. And then RAG1's partner, RAG2, does sort of the next step of this. And so you can see this book is saying it's RAG1, RAG2, and I don't care if you could say RAG proteins do this. I don't care if you know which RAG1 or 2 does each of these steps. It's just I feel like I have to be technically correct when I say it. RAG2 then does its enzymatic activity. So RAG2 has an important job in this whole process, which is that RAG2 is going to do something called nick the DNA. It's going to form something called a DNA nick. First, I want to mention a couple of things about the way the diagrams are going to be shown on the next couple of slides. And then I'm going to get back to telling you about the DNA nick, including defining what a DNA nick is. So what I want you to notice here, because they already do this, is if you note, we've got our V and our J segment, and then they start just showing us one segment. They just show us the V here. There is also a nick happening at the J at the same time. So there should actually be two nicks, a V nick and a J nick. But what they usually do at this situation is they usually kind of simplify the drawing and only show us what's happening at one spot instead of showing us both of them all the way through the figures, because it would just be like way too confusing and complicated to be showing us both of them all the time. Sometimes we're even going to only talk about one of the DNA strands and not even mention what the other one is doing, or not even like draw out the sequence. Like I'm going to actually draw some sequence at one point, and I'm just going to be drawing this one top strand. I'm not even going to draw the bottom strand. Again, for simplicity's sake, at least you'll see what you'll see. But know that actually this is happening at two places at once. The two segments that are getting joined together. But if we tried to put all of that on one slide, it would be a giganto mess. So yes, we're only going to be showing you this happening at one mini gene site. So here you can see it happening at V, but it is also happening at the same time at the other site that's getting joined. It's just hard to depict that for you. So like I said, RAG2 performs this function of making a DNA nick. And a DNA nick is a specific molecular biology term of what happens here. So a DNA nick means that the break that you're making in the DNA is a single-stranded break. So it means that you're breaking one strand but not the other. So when I say RAG2 nicks the DNA, that means it's breaking one strand but not the other. And you can see that single-strand nick here. You can see that we get this break on one strand, but the other strand is not broken as part of our nick. And so now you can see our nick again at the top. And if you remember details of sort of general DNA structure, remember that each base pair has that's the wrong screen, has a phosphate, and then the base, and then the OH, and that binds to a phosphate and a base and the OH, right? Or like 5' phosphate, A, OH is bound to the phosphate of G, and then there's OH, right? This is my very intense chemistry that we're seeing here. So you see how we have those phosphates and those OHs bound together on either side of the base. When RAG2 makes the nick, it makes the nick between the RSS and the gene segment. So it actually specifically nicks right at the beginning of the heptamer. So you can see that it's making that nick right between the gene segment and the triangle. It's like exact on the exact spot. It's making a single strand break there. And when that break happens, as you could sort of imagine from my very complicated chemistry that I've drawn for you, you end up with a free 3' hydroxyl where you had the break, and you end up with a free phosphate. So basically you've broken this DNA, and so whichever base is here, still the wrong screen. Wonder how many times I'm going to do that today. You have your free hydroxyl here, you have your free phosphate here. They used to be bound together. So we've got those, you can see this at the bottom. We've got this free hydroxyl, free phosphate that are made because of that nick. The hydroxyl does not want to be free. Sad for it. I want to be free. The hydroxyl does not want to be free. The hydroxyl would like to be bound to something. And so, are you ready for perhaps the most chemistry of the semester? The free hydroxyl, the OH, is going to perform a nucleophilic attack. Like I said, it's hardcore chemistry going on here. That hydroxyl is going to perform a nucleophilic attack. It wants to be bound to a phosphate. And the nearest phosphate that it can be bound to is the one on the other strand across from it. And so you can see this hydroxyl is now going to bind to this phosphate down here. And so you can see that now you're going to have the part with the RSS, which is here in yellow, basically have a double strand break at the end. And you're going to see the part that with the gene segment is actually going to sort of, the top strand's going to loop around to continue into the bottom strand. It's called a hairpin. And so you can see we get this nick formed. We get the part with the RSS, sort of just with this free end of double-stranded DNA. The RSS, if you remember, is the signal. So sometimes we call this the signal end because it's the rando piece of DNA containing the signal. I've also heard, for some reason, I learned it as the signal joint or the signal join. So sometimes you'll hear me say the signal join. I mean the signal end, all the same thing. And then we also have this one that has the V part. It's the part that's eventually going to code for the antibody. So it's called the coding end or the coding joint or the coding join. And so you can see we've now basically broken the DNA exactly between the gene segment and the RSS. The part with the signal, which is honestly the trash part, has this double strand, like normal looking double stranded DNA. And the coding end, the part that we're going to use, has this weird thing called a hairpin, where the top strand actually then can sort of continues itself down to the bottom strand. Yep, Chrissy.
So is RAG still there preventing the 3' hydroxyl from reassociating with the...
So it's an interesting question. Probably, but also I could imagine that there are situations where it reanneals and then we just never see it. Like we would never see that cell because that cell would like go on about its life having not done VDJ recombination. So I can't tell you like how frequently that happens versus the hairpin, but versus this nucleophilic attack, because we actually could never measure that cell that did that reannealing. So we've got our nick formed by rag2, and then we've got our hairpin. And so now what we need to do is we need to sort of hear the rest of the story of the signal end and the rest of the story of the coding end. So we've got two different players here who have their own story. And if we're being realistic, we actually have four. One coding end from V, coding end from J, signal end from V, signal end from J. But we're only drawing two of them here to make our life easier. So we're first going to hear the story of what happens to the signal end, the signal joint. It's the trash part. It's the part that we're not actually going to really care much about again. It also, and so part of it is like I'm going to tell you what happens to that trash DNA and then we don't have to worry about it anymore. It also is actually a little bit of the simpler one of what happens to it. So that's why we talk about it first. So you can imagine we've got this signal end that came from V. This is our RSS. See, it's still yellow. It has just a normal double strand end of DNA, not this weird hairpin business. And so you got two of these normal old double strand DNA ends with signals from the two different gene segments you're combining. And you ligate them together. You take those two ends and you just paste them together. And so that's pretty much all that happens with a signal joint. And so here you can see rag1 and rag2 are doing this reaction at both the V and the J. So you can see you've got two hairpins and two signal joints. And then, you know, that yellow signal joint and that brown signal are just going to get glued together. And we're all going to live happily ever after. They're going to get glued together perfectly. It's just DNA ligase. You can see examples of this from your textbook, which sort of show it in slightly, you know, more detail in terms of who the players are. But it's normal DNA repair proteins. Normal old ligase just finds these two double-stranded pieces and joins them together. Notice that all of these are showing sort of rag as like a blue blob. And that's really the rag proteins together in a complex. And so notice, and this will actually come back later, rag is sort of holding on to these ends, to all of these four DNA ends, and not letting them like float around willy-nilly. And so that's why the two signal ends can easily get joined together because they're both being held right next to each other. And you just paste them together. And then we still have our two coding joints that we need to talk about. Yep, Julie.
The figure on the other side that's also in here, like
Mm-hmm.
small, with
Yep.
the coding end and the signal end.
Mm-hmm.
Why doesn't the hydroxyl then do another nucleophilic attack on that phosphate and like make another hairpin? Like, can it not?
I think it's because of how what rag is catalyzing. And I think that also it's because, yeah, I think it's just how rag is holding them is my best answer for you. Okay. I hadn't really thought about that, but I think it's more of just like what rag allows or doesn't allow in terms of chemical reactions. Okay. So we're basically just going to paste together our signal joints. And then we've got this circle of DNA. And you can see that circle of DNA. We don't do anything fancy. And we throw it away. And no one cares. The more complicated thing is what the heck do we do with the hairpins? You've learned about, you know, this general idea of you take a double strand DNA and you paste it together. That's sort of normal DNA repair. But you can't just paste the hairpins together. They're already like top strand bound to the bottom strand. They're not just going to attach. You're going to make some wackadoodle thing if you did that. So you actually need to deal with the hairpin before you can paste the two coding joints together. And so the sort of interesting part of what happens here is that we have to figure out how the heck do we deal with those hairpins before we can paste these two things back together, because the hairpin just can't be pasted as it is. So that's just sort of, that would be an interesting kind of molecular genetics-y question just in and of itself. But the reason why we as immunologists find this particularly interesting is that when we deal with the hairpin, we can sometimes add and subtract base pairs. And so this dealing with the hairpin is how we get junctional diversity. It's how we get the additional base pairs that we see in the coding joint. And so that I would say the phrasing should be the other way. Variation at the coding joint leads to junctional diversity. Basically, because we don't do the hairpin perfectly, because we add and subtract base pairs when we deal with the hairpin, that's why when we put together our two segments, our V and our J, we might end up getting different products in different B cells, because those different B cells might have dealt with their hairpins slightly differently, and that led to some addition and subtraction of base pairs. And so the reason why we kind of have to go through this, even though you're like, why is she doing molecular genetics today, is because we wouldn't have junctional diversity. We wouldn't have all of these great diversity mechanisms if we didn't have these processes for dealing with the hairpin. All right, so here's our hairpin. RAG2 nicked the DNA. We made this hairpin at the coding end. So let's imagine, first of all, that you were a person, you were a patient who could not open the hairpins. So let's imagine that it's a cell that cannot open the hairpin. Okay, first. What happens if you can't open this hairpin? What happens if you don't have an ability to deal with this hairpin? Yeah, David.
That piece of, or assuming this like the part of the chromosome, that entire, you have like literal break in the chromosome, so the cell might not be able to mitose correctly and will cause a lot of problems.
Yeah, you're just going to have random broken pieces of DNA in the cell, right? You are not going to be able to, if you can't open that hairpin and get stuff fixed, that cell's probably going to die. This is a developing B cell or a developing T cell. What happens if your developing B cell or developing T cell dies because it can't open the hairpin? Yeah, Jazz. Yeah, this would mean you make no B cells and T cells. This would give you a total loss of the adaptive immune system, total immunodeficiency, if you can't open this hairpin. And so if we had a patient who could not open the hairpin, they would die as a child of infectious diseases because they would not have B cells and T cells. Okay? That's going to come back. You'll see. So if you were in charge of all of this, how would you deal with the hairpin? What would your answer be in terms of how you deal with the hairpin? What do you think should be the answer of deal with the hairpin? Yeah, Paige. Mm-hmm. So just break it, right? Yeah, just break the DNA. Just have an enzyme that cuts the DNA or breaks the DNA, right? When I was in your shoes as an undergrad, the answer was, and then the DNA is broken. And we didn't actually know how the DNA got broken. We just knew it got broken somehow. But nobody knew the details of it. We knew there was some fishiness, and we'll get into the fishiness of the breaking. But we just know, and then the DNA is broken. In my first year of grad school, somebody discovered the enzyme that did the breaking and discovered the enzyme that does this situation. So if you think about it, that enzyme helps you survive childhood and not get infectious disease, right? It like protects you as a child. Pretty nice of that enzyme. Thank you, enzyme. And so the people who discovered this enzyme decided to name it after the Greek goddess who protects women and children, because it's protecting children from being immunodeficient. And that Greek goddess is named Artemis. And so that's how Artemis got its name, is that it was the Greek goddess who protects children, because it was protecting children from having an immunodeficiency. And Artemis is the enzyme that, as Paige points out, cuts open this hairpin. Artemis is great. Artemis also has one kind of thing that it kind of sucks at its job. One little thing about fishiness going on with Artemis. So you can see a hairpin at the top, and this is sort of drawn out on this slide. I'm going to draw on the board and some sequence that I hope will make it better. In the textbook, they often show like 2 base pairs of things. I usually draw more than 2 base pairs to make it kind of easier to see. Okay? So there's my hairpin. Okay? This is the bond that came as a result of the phosphodiester. And I draw it here as sort of a big, like, curvy bond. But I'm going to redraw it for a second. It's just like a little phosphodiester bond. Okay? That's a phosphodiester bond. That's a phosphodiester bond. That's a phosphodiester bond. That's a phosphodiester bond. That. Actually, those are phosphodiester bonds too. Look at all those phosphodiester bonds right in this area. Artemis is awesome because it knows how to cleave the phosphodiester bond, but it doesn't know which one to cleave. Huh? What?
That's terrifying.
So Artemis doesn't know which it knows it's supposed to cleave a phosphodiester bond in this area. It's being held as part of a complex, as part of the blue blob in this area, but it doesn't know which phosphodiester bond to cleave. It can cleave up to 5 bases off in either direction. So it could cleave here, 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 here. It could cleave any of those bonds because it's just cleaving a phosphodiester bond around here. It doesn't know which one. Yep, Julie.
Sorry, other thing. The
Yeah.
T.
Mm-hmm. Oh my goodness.
Okay, no, I just want to say.
Thank you. This is called What Happens When Dr. Barker Doesn't Get Enough Sleep.
No, I didn't know
Yeah.
there was like
No,
something else going on with
no,
that, or- okay.
not enough sleep is what's going on with that. Sorry. So Artemis can cleave in all those places, 5 base pairs in either direction. So first, let's imagine that Artemis picks correctly. So Artemis picks this bond right here. What is the result if Artemis picks that bond? Yeah, Paige. You got 2 strands that are the same amount of base pairs. You got a nice little double strand piece of DNA. Easy to work with. Nothing changes. Nothing exciting happens. No one cares. Well, we do care, but just whatever. And in fact, it's showing us as number 2 in the example on the slide is where Artemis cleaves in that correct location. And we get product number 2, just normal looking DNA. But now let's imagine Artemis cleaves there. Again, I draw more, your book only does it with like 2 base pairs. I do it with slightly more because I think it's easier to see with more base pairs. So what happens now? What product do we get? Yeah, Paige. Okay, what strand's extra long? The bottom one. All right, what does the bottom one look like? Do people see how Paige got that? She basically was reading this strand and then looped around and read until the site where it was cut. And that is totally correct. What's the other strand look like? So if I'm starting, oh my God. What? What's the other strand look like? Now that's what I get if Artemis cleaves 5 base pairs in one direction off. And in fact, this image from a textbook is showing you either site 1 or site 3, where it could either cut too far to the left or right, or too far 5' or 3'. And so you can see you might get the long strand at the bottom, you might get the long strand at the top, whatever. Now we got a problem. We can't take these different pieces of DNA that have like weird raggedy ends and ligate them together. We can't really ligate together our raggedy ends. And so, fortunately, the blue blob complex that you saw on previous slides, there's this protein complex that's just labeled RAG and it's a blue blob and everything. There's some members there. And one of the members is polymerase. Just general old polymerase at this point. Just general DNA polymerase. And when polymerase is around, which it always is when this is happening, and sees this situation, polymerase is like, ooh, 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 me, I know what to do, I know what to do, I know how to deal with this, I can handle it, I can handle it. I, polymerase, know what to do. So now, what is polymerase going to do here? Yeah, Emily. Fill in the top strand. So what's it gonna do?
Oh.
It's okay. Do you see how many have I gotten wrong today? I can't judge anybody.
T, C, A, G, A, T.
Awesome. All right. So now let's compare what the chromosome looked like before, sort of the before DNA, and the after DNA. This is the after, this is the before. What do you notice as we're comparing our before and after DNA? What the heck did I do wrong? Did I do another one wrong? TACA G. Yeah, I was like, no, I think I flipped those two. I think this is supposed to be an A, and this is supposed to be a T. T, yeah. A, oh, you're right, okay. God, Lord. Okay, oh my goodness, I'm so sorry. Yes, Jazz?
It's correct until the part where that phosphodiester bond that was supposed to be cleaved wasn't cleaved. So now you have 4 that are incorrect.
So I basically have the original DNA and 4 new base pairs. So I basically rebuilt the original and added 4 things that were not in the original genome.
Because this lecture didn't seem cursed enough, at this point the microphone died, and so I'm now having to re-record the rest of this lecture at a later time. I want to talk through a little bit about what I was saying about P nucleotides using the information on this slide, since you weren't able to see the information that I was writing on the board. And so what we can see is our hairpin that has happened, that is here, and it could be cleaved at 3 different places. It actually can be cleaved at slightly more than 3 different places, but we've got 3 examples here: site 1, site 2, and site 3. If you look at site 2, would be a cleavage at the exact spot, the exact phosphodiester bond that was made at the hairpin. And so when that happens, we would get the blunt-ended product that's shown in the middle. It's basically the same product that we had before the hairpin, if there was no hairpin that had happened and we just made a double-stranded break. That one's actually not very interesting, so we're not going to talk about that one too much. If, however, we cleave at sites 1 or site 3, you can see that we get products that have an overhang. If we cleave at site 1 near the top strand's a little shorter, the bottom strand's a little longer, because some of the DNA that should have been part of the top strand is now part of the bottom strand. If we cleave at site 3, you can see that's towards the bottom. Now the top strand's a little long because it has extra material that came from the bottom strand. The bottom strand is a little short. And as I was saying right before this, we can't ligate together the raggedy ends of the DNA that happen either when we cleave at site 1 or at site 3. And so we have a polymerase that will come in and fill in those ends. And you can see this process happening at the bottom of this slide. So if you look at the bottom of the slide, you can see an example with this V region where you have cleavage that looks sort of like the cleavage above at site 3. And now we can actually see the base pairs TC on the top strand and the AG that is paired with them on the bottom strand. And we can see the hairpin. And you can see a situation where the hairpin is cleaved on that bottom strand right after that A. And as a result, you can see that the top strand gets a little longer. It's got TCGA. The GA came from the bottom strand because the 2 strands were attached. And you can see that the bottom strand is now a little short. So that's that raggedy end that I was talking about. And polymerase will come in and fill in that gap. And so we'll end up with this product that we see here, where we have TCGA and we have AGCT. We're filling in this gap that had been left previously from hairpin opening. And you can see it's just complementary base pairing filling in with polymerase. What we can notice is that if we look at what the original DNA that we had in the genome, that's in every other cell, that was present before all of this, we look at that, what that DNA is, it's this TCAG and all the other stuff. And we have 2 additional base pairs, a GC base pair and an AT base pair, that have now been added, that were not part of the original genome. They're not present in any cell other than this one. And this came as a result of the hairpin being opened unequally. So we've now added 2 brand new base pairs that aren't from the original genome. You can see the same kind of thing in the example with the J segment on the left. So you can see that there is this ATTA base pair. You can see now this is a little bit like situation number 1 at the top, where we have cleavage leading to this asymmetric product where we have a longer bottom strand. We've got a gap in the top strand, and polymerase will come in and fill in that gap to give us the product that we see at the bottom. And so you can see that product here. And again, we've added in 2 base pairs. They're shown here in blue that were not part of the original genome. They're not present in any cell other than this one that did this type of hairpin opening. And so these are 2 novel base pairs. If we look at the DNA, we can also notice something else kind of interesting. If you look at this section on the V region that you see here, if we read the top strand from 5' to 3', we read TCGA. If we read the bottom strand, 5' to 3', we get TCGA. It's identical to the top strand. Same thing if we look at this other strand and we read from 5' to 3', we have TATA on the top. We read 5' to 3', we have TATA on the bottom. So these sequences read the same forward and backward. In English, the term for something that reads the same forward and backward is called a palindrome. And so these nucleotides that are added, the 2 in blue here or the 2 in blue here, are known as P nucleotides or palindromic nucleotides. So P nucleotides are the result of unequal hairpin opening and then filling in by this polymerase. They end up being palindromic because they came from the other strand, and so there is some complementarity within that sequence. And you can see this process happening here as well. So you can see we get this nick between the D segment and the heptamer right here. Also between the J segment and the heptamer right here. And we end up with our hairpins that you can see in the middle image. You can see the D segment will be cut at the bottom. The J segment will be cut at the top. And we can see that we get this uneven DNA, which now can be filled in by polymerase. And we now have this additional DNA that was not present in the original genome, which would be called the P nucleotides. This comes from hairpin opening. You can watch this process happening here. So here I'm giving you a variable region from a light chain and a joining region or a J region from a light chain. You can see I've got the heptamer, the spacer, and the nonomer sequence attached to those. And the black nucleic acid is the V region or the J region and is the actual DNA that's in the germline, is in every other cell in the body in this organism. So the first thing that will happen is that RAG2 will make a nick. And so you can see that nick here. We have a free 3' hydroxyl because of that nick. And that 3' hydroxyl performs a nucleophilic attack on the base pair across from it, which you can see here. And you can see that that happened right between the gene segment and the RSS. The result is that we get this hairpin. And now that hairpin can be cleaved in one of many possible sites. So let's assume that our hairpin cleaves right here. If the hairpin cleaves in that spot, then we're going to eventually get a T down here, then an A, then a T, then an A, then a C. Added. This section will be empty, and then will be refilled in by polymerase. You can see that polymerase will also add in some additional base pairs. And so you can see now that the things that I have written in red would be the new nucleotides that were not present in the original germline sequence. Those would be the P nucleotides that came from our unequal hairpin opening. And so this is where we get those P nucleotides from. There's one other way that we can get new nucleotides added to give us more junctional diversity. This slide also shows some nucleotides being removed by exonucleolytic cleavage, which is true, but which I'm not going to be asking about on any exams. So I don't want you to get super focused on it, but it is true that there is exonucleolytic cleavage at this point. However, the thing that I want you to know about is that there's this other polymerase that can act at this point. This polymerase is known as TDT. And TDT is a very special type of DNA polymerase because it is able to add base pairs without a template. So that is very different than other DNA polymerases that you will have learned about. TDT doesn't need to have something to base pair with in order to add new nucleotides. It just adds whatever nucleotide it feels like. Finds a T, it adds a T. Finds an A, it adds an A. Adds whatever it wants. And so TDT can also just add some random nucleotides because it feels like it. Because they're random, we refer to those as N nucleotides. And so we do have some N nucleotides that get added. We do have some polymerase that's going to come in and clean up this area. And then at the end, we're going to finally ligate together our D and our J segments. You can see now that there's this new DNA, P and N nucleotides, in between the D and the J. And those new nucleotides were not in the original germline sequence, but have been added by these junctional diversity processes of P and N nucleotide addition. So if we think about what's happening with the coding joint, we have our DNA ends that are bound. We've got hairpins. The hairpins will be opened by Artemis. That may add some P nucleotides. TDT will also come in and act. That can add some N nucleotides. And then we'll ligate together this product and eventually make this coding joint where we will have that red segment and the yellow segment that we started with, that are our mini gene segments that we're joining together. But you can also see we've got some additional nucleotides that have been added right at that joint. So I want to show you a couple of different questions I have asked in the past about this so that we can walk through how you would do these questions. And so here you can see I'm telling you about a B cell, and I tell you which V, D, and J segments it uses. And then I zoom in on two of those sequences that would have been joined together. In this case, the D and the J of the heavy chain. And I show you the germline sequence, basically the before sequence. And then I also show you the after sequence. And in the question, I ask for you to label the nucleotide, the origin of the nucleotide. So I want you to know where those nucleotides came from. There are about 4 parts to this. You can see it's worth 4 points. And so each part gets about a point, though I have scored it slightly differently in different years. And so the first part is actually pretty easy. The first thing that we can do is we can look at the DH2 segment in the before, and we can look at the after, and we see that there are base pairs that just came from DH2. They're just the actual DH2 segments. That's easy. That was their origin, was they were in the genome as DH2. We can also look and we can see that there are some base pairs that were part of JH3. that had their origin as part of JH3 in the germline. But now we have these 4 base pairs remaining, and we need to figure out where these 4 base pairs came from. And these 4 base pairs could potentially be P nucleotides or N nucleotides. So basically what we need to do, is we know that they weren't in the original germline, they were added, but we have to figure out if they are P nucleotides or N nucleotides. And we're going to determine this one by one for each of these nucleotides. So we're first going to look at the first A. And so here, we're gonna look at this A. If this A had been a P nucleotide that originated from the DH2 segment, it would have been base paired to that T originally. It would have been a hairpin that had been cleaved in the wrong spot. That's totally possible. A does bind with T, and so that base pair could be a P nucleotide. We can then move on to the next A. If that A had been a P nucleotide, it would have been bound to this A on the DH2 segment. That's not right. A doesn't go with A, so that can't possibly have been a P nucleotide on that side. If you remember, P nucleotides are added before N nucleotides. So if we get to a point where there's no P nucleotides being added on one side, we can look at the other side. So now we can look at this C. If this C was a P nucleotide on this side, it would have been generated by having been bound to this G next to it. That's totally possible. So this can be a P nucleotide. Next, we look at this G. If that G had been a P nucleotide, it would have been bound to that base pair, which is a G in this case, and that's not possible. So we know that couldn't have been a P nucleotide. So now we have these 2 leftover nucleotides. They must have been N or random nucleotides that were added by TDT. And so the way I would want you to answer this question is to indicate which sequence was DH2, which sequence was JH3, and then for the original other base pairs, I would want you to label them as P or N. And you can see a similar question here. At this point, I got a little smarter with writing the question and say, if the nucleotides could be P nucleotides, you must label them as P nucleotides, thanks to a clever student argument in a past year. And so the first thing that I can do is I can label my germline sequences. So now I've done so, and again in this problem I have 4 extra base pairs because I'm apparently not very creative. So I'll first look at the G. If the G was a P nucleotide, it would have been bound to this A that is to its left. That's not possible. So I'm already done with the left side. Now if I look at the right side, the T could have been bound to the A. That is correct, so that's a P nucleotide. Next, I'm going to go to this A. The A could have been bound to the T, so that is a P nucleotide. Now I'm going to keep going. Could this G have been bound to the T? No, it could not. So that must be an N, and this must also be an N. And so again, this is the way that you would answer a question like this. And so this is the overall process of VDJ recombination. We see V, D, and J segments joined together in order to make the variable region of the heavy chain. And we see V and J segments put together in order to make the variable region of the light chain to give us our very diverse antibodies. The isotypes, IgM, IgD, IgG, IgE, and IgA, are encoded by different exons of constant region information. So you can see the C mu constant region just downstream of the VDJ gene segments, then you can see C delta, then you can see the C gammas for IgG, then you can see C epsilon and C alpha. What you can notice is that the mu constant region is relatively close in terms of the amount of DNA between the VD and J segments and that constant region section. There's also a pretty short amount of DNA before we get to the delta constant region. But you'll notice there's a very long stretch of DNA between delta and all the rest of the constant region genetic information. And so when we get to the point of transcription from this region, we see our polymerase, our RNA polymerase, basically only transcribe the VDJ, the constant region for IgM, C mu, and the constant region for IgD, C delta, before basically getting tired and not being able to make it across that whole rest of the 54 kilobase sequence. And so when we look at what the RNA looks like from this situation, we will see we have an RNA that has the VDJ join region. You can also see that RNA here. And then it has the information for C mu and C delta. The DNA is identical in either situation, but that RNA can be spliced in two different ways. In one way, it can be spliced to add the mu constant region information next to VDJ. The other, it can have the delta constant region. And so our cell is actually able to make both IgM and IgD at the same time. Right away after that VDJ recombination, but it can't make any of the other isotypes at this point. Our B cell receptor is also coming from this same location. Again, we've mentioned that the B cell receptor is an antibody with a transmembrane domain, and you can see my C mu region, my C delta region in the RNA that I've mentioned on the previous slide. There also are ways that those RNAs can be alternatively spliced to either make a version that ends right after the C mu and thus is a secreted protein, or to make a version that has a transmembrane domain and thus is a B cell receptor. And so the reason why we can have both the antibody and the B cell receptor is due to alternative splicing of the RNA product. It doesn't require any changes to the DNA. One thing to note about both the production of IgM and IgD that I showed you on the previous slide, as well as the production of secreted antibody versus membrane-bound B cell receptors that I show you here, is that in both of these situations, we're looking at alternative splicing of RNA, and we're not looking at a change to the DNA. And so with the alternative splicing of the RNA, the cell could make multiple RNAs from that same DNA and splice them differently, and could be making two things at once. This cell could mostly make B cell receptor, or mostly make antibody, or mostly make IgD, or mostly make IgD or IgM, and kind of be switching back and forth between all of these things. None of these changes are irreversible because they're changes with RNA instead of changes to that original DNA. If you change the DNA, you can't go back. But if you're making changes to RNA, that is a reversible change. We talked previously about the danger that is inherent in VDJ recombination and the fact that sometimes we can see recombination errors that can lead to a number of issues like the leukemias and lymphomas shown on this slide. And so we have to think a little bit about how this process happens in order to limit recombination errors. One of the ways that this works is shown in both of these images. And as you look back at images of this process, you often see the RAG proteins just shown as like a blue blob that says RAG. It doesn't indicate RAG1 versus RAG2. It doesn't show you Artemis or TDT or anything like that. And the reason here is that all of these enzymes are going to act together as a complex. They're basically all bound together and acting together in one big clump, shown here as a blue blob. What that means is that they are able to hold the DNA ends and not let those DNA ends start to move around the nucleus and potentially interact with other DNA. We're not going to see chemical reactions with other random pieces of DNA because RAG1 and RAG2 are holding all of the DNA that we're working with very close together. So if there's going to be some kind of reaction, the easiest reaction is just to react with the DNA that's held right next to you. And so by keeping those DNA ends from invading other chromosomes and making sure that all of these reactions are just happening on the DNA we want, by holding all that DNA in a complex together, we can really limit many of the recombination errors that we might see. The other really big way that this process is regulated is that there is extremely tight regulation of expression of the RAG genes. So every cell in your body has the DNA to potentially transcribe and translate RAG, but every cell of your body does not make the RAG enzymes. They usually just leave that DNA alone, don't transcribe it, thus don't have anything to translate. The only cells in your body that turn on RAG are developing B cells and developing T cells. And you can see this here. You can notice that at the beginning, RAG is off in this stem cell that's getting ready to become a B cell. You can see that there's a period of time where RAG gets turned on, a period where it gets turned off, and then it gets turned back on. Then RAG is off after that, and RAG will be off for the rest of the life of the B cell. There's something similar that happens with T cells, where basically there are 2 times where T cells turn on RAG, and after that RAG is off forever, and every other cell in the body keeps RAG off. And so the fact that RAG is really tightly expressed to only be turned on in the cells that need to do VDJ recombination at the stages of their life when they need to do VDJ recombination, and it is never turned on any other time, is really key to making sure that we don't see damage to other parts of the DNA. So all of these processes that we've been talking about involving different pieces of DNA and RNA are of course happening inside a cell. Specifically, this whole process is happening inside a B cell, and that B cell may go ahead and make antibodies. And this is happening when these B cells are developing, when they're sort of sequestered or sheltered in the primary lymphoid organ, in this case, the bone marrow, where you can imagine that there are absolutely no pathogens. And we basically see that our stem cells undergo VDJ recombination away from pathogens, not trying to do recombination to make something that matches a particular antigen, but basically trying to do recombination to make a full breadth of different cells. Then after we've done that sequestered, secluded process of making all of the diverse B cells, those B cells are released to the rest of the body, where they might find antigen and where they might be able to proliferate, make many more copies of themselves to be able to act against a microbe. And so VDJ recombination is happening here in our response long before we're actually making any response to antigen. We do not do VDJ recombination in order to try to match an antigen. We already have done it to have a receptor that can respond to every possible antigen. There are some other kinds of changes that can happen to the antibody genetic material. One of them is that we can actually have a switch of which constant region exons we're using to switch from maybe using IgM to IgD. We can also see some changes in the site that binds to antigen to influence the affinity of the antigen. That change in affinity in terms of the change in the protein is called affinity maturation. And the change in the constant region from IgM to IgG or to some other ones is called class switch. Both affinity maturation and class switch involve changes that are made to the DNA, so they're not going to be reversible, while the changes we saw before to the RNA are reversible. Affinity maturation actually involves a DNA change called somatic hypermutation, which you can see listed here. But what I want you to know is that both of these things happen much later in the life of the B cell. These are not things that are happening in the bone marrow, and they're not things that we need to worry about right now. What you might also guess from what I'm saying here is that these activities do not involve RAG because they are things that do not happen in the bone marrow. They happen later in the life of the B cell when RAG is totally off. And so if we want to use some of those constant regions for the other isotypes like IgG, IgE, or IgA, then we're going to have some changes happen at the DNA level because those isotypes are so far away that we are not going to be able to do transcription of them originally. So early on in the life of the B cell, it only has IgM and IgD as an option. Otherwise, that B cell will have to undergo some other changes, again, that are happening later in its life, not at the part of its life we're thinking about right now. And so this slide just sort of gives you a summary of some of the changes that happen to the immunoglobulin genes. You can see that we have the recombination of DNA and generation of junctional diversity, which are irreversible changes because they're changes that are happening to the DNA. We can do things like make IgM versus IgD or membrane antibody versus membrane B cell receptor versus antibody. Those are happening because of RNA splicing, so they are reversible. Later on, we'll see some other changes that will happen to our immunoglobulin genes, and they are going to be irreversible changes since they're happening to the DNA. And you can see this is another table just summarizing some of the things that we know about that are important in B cell diversity, which may be useful to think about when we look at T cell diversity later as a compare and contrast. Thanks.